It is now my great pleasure to introduce our keynote speaker. Our keynote speaker is Floyd Romesberg. He's coming from the Scripps Research Institute, and he's a professor in chemistry. He will be talking about synthetic biology and semi-synthetic organisms. So again, to put the balance here between the CRISPR discussions and synthetic biology, we're looking forward to your talk, learning more on what is it that we can do today and what is that we can dream of eventually doing in the future. Thanks a lot for accepting the invitation.
Is this good? Can everyone hear me? Or too loud? Or it's good, or turn it up? Okay. I'd like to thank Catalina for the opportunity to come and tell you about some of my group's research at the Scripps Research Institute in San Diego. It's a real pleasure to come here. It's always a pleasure to visit. Vancouver's a really great place. I enjoy it. What my group's interested in is, so let me first give you sort of a disclaimer. I'm a chemist, so I was trained as sort of a chemist, in fact, I was sort of a physical chemist. So a lot of what we do in my group really draws from a sort of a very chemical approach. And what my group's interested in is largely evolution and thinking about evolution from a variety of different perspectives, like I said, largely chemical. And so what I'm going to talk about today is our efforts to develop an unnatural base pair with which to expand the genetic alphabet and eventually the genetic code. So let me start by talking a little bit about synthetic biology and how we think about it. And so I'm a chemist, so synthetic means something to me. And most synthetic biology that gets done today is rearranging natural components into novel functional units. To us, synthetic biology, the opportunity provided by this new field is to use the power of chemistry, the power of synthesis, to come in and build parts that are not native to cells, to life, and try to integrate them into the biological systems of life. And I think there's a variety of reasons you could do that, or that you'd be interested in that, because you can sort of probe these sort of interactions and the sort of behaviors that are mediated by the natural systems. And it also might give you the ability to expand it more by increased orthogonality. And so if you want to talk about what the broader notion of what synthetic biology is, it was originally defined about 100 years ago by Stephen Leduc as the effort to create novel forms and functions. So if you want to think about novel forms, novel life forms, novel functions, the functions those life forms can perform, it's interesting to think about how nature does that. And of course, nature does that by encoding all the information of life in a four-letter genetic alphabet, G, C, A, and T, or U. And those four letters form two base pairs, and those base pairs are what mediate the stable retention of that information via replication. And then they also mediate the stable recovery of that information, the use of that information through transcription and translation. So it's the selective pairing of G with C and A with T via those hydrogen bonding interactions that mediate, that allow for the stable genetic alphabet. So just, I think everyone here knows, here are those nucleobases that differentiate G, C, A, and T, and I'm not showing you here. And of course, each of them are attached to a sugar and a phosphate backbone. And this is that piece of DNA. And if you line two of them up, they form these H bonds. One strand runs this way, one strand runs that way. And that's, of course, why DNA is stable. It's replicated by splitting them apart. A DNA polymerase coming in and replicating them based on recognition of those hydrogen bonding partners. So when I started my lab about 15, 16 years ago, one of the questions when we were thinking about this was, in getting back to this issue about building synthetic parts and examining, learning something about the natural biological parts or biology in general, we asked, obviously hydrogen bonds are important for this code, this four-letter two base pair code, but is complementary hydrogen bonding the only force that would have been suitable for information storage? And so the goal in our lab was to expand the genetic alphabet by creating two new letters that pair with each other to form a new base pair that functions alongside with G, C, A, and T. And to mediate those base pairing, to examine a different force, we thought we would examine, we would try to use the hydrophobic effect, the fact that oily molecules want to get out of water, and that oily molecules like to pair with each other And then within the duplex, they would also, specificity would be mediated by packing interactions with each other and with the flanking nucleobases. And so just a quick couple words on why we'd want to do this. Why would we want to expand the genetic alphabet and the genetic code? Well, as I just mentioned, I think there are profound and important conceptual issues here. I think everyone sort of assumes that hydrogen bonds, I mean, it's not like we believe in the vital principle anymore, but I think people in general still somehow think that biology is different than non-biological systems. Maybe purely by complexity, maybe purely by the fact that evolution's had a 2 billion year head start on us. But if we could take totally hydrophobic interactions and expand the genetic alphabet, that would tell us something about evolution maybe, in that hydrogen bonds weren't the only force, they're simply the ones evolution used. And so I think as a chemist, that was something that we were very interested in. Protein therapeutics. You have to have lived under a rock if you haven't noticed the revolution in protein therapeutics. The last data that I've seen was from a couple years ago at the FDA, where 70% of the INDs, the investigational new drugs, these are the drugs that are coming down the pipeline, 70% of them are protein therapeutics. Antibodies, cytokines. Now, the properties that protein therapeutics can have, the diseases they can be used to treat, must in somehow be related to the properties of their constituent amino acids. And there are only 20. To a synthetic chemist, to a medicinal chemist, that's weird, right? Because if you look at small molecule medicinal chemistry, the single pharmacophore that's been most useful is an electrophile. And a lot of drugs have them, yet there is no electrophile available to a protein. So what would happen if you could give a protein an electrophile with an unnatural amino acid? Could you create new protein therapeutics? And so there's all sorts of, I think, the potential to expand the utility of protein therapeutics was something that has really driven us. And then just the idea of getting back to Leduc's definition, the creation of novel forms and functions, I think, is a really interesting challenge. So I'm not going to go into the chemistry. My last spent 15 years, the program very much proceeded like a medicinal chemistry program. We wanted to examine hydrophobic analogs, so we started by making nucleotides that simply had benzene rings. And and then after about 15 years where we simply added modifications, we had activity assays, mostly DNA replication, where we created structure-activity relationships, used that to feed back into the design. We made over 200 nucleotide analogs and analyzed them. The ones that we finally found, we also supplemented that effort with 2 independent screens. The first pair that we found that actually sort of worked was this pair. So the name stands for the methyl-methoxy, the second of a methyl-methoxy series that we had made. And this 5,6 is this 5-methyl sulfur isocarbyl styrol. That's what that chemist calls that particular scaffold. So don't worry about the names. It's kind of silly, but you give me better ones and we'll use them. And so this was the first that we had that was actually replicated well. And I'm not drawing the sugar and the phosphate, but of course they're there. I'm only focusing on the nucleobase portion. And very quickly we then identified this naphthyl methoxy derivative that was a better partner for 5,6. And then this thiophene derivative that we call TPT3 that was a better partner for NAM. So this iterative walking by improving one partner, then improving the other. Really led us to the first unnatural base pairs that could PCR amplify well, with high fidelity, high efficiency. And so then that motivated us to try to take the system and deploy it in a living cell. That has always been the long-term goal. So I don't have a lot of time to describe this. If one's interested in getting triphosphates into a cell, that's a problem. And so the literature will suggest a couple of methods, and if any of you want to wonder why we didn't, want to talk about why we didn't use those methods in the end, I'd be happy to tell you. Short story is they don't work in bacterial cells. This is all an E. coli cell. Maybe you can see 2 membranes there. What we wound up working was stealing a dedicated nucleoside triphosphate transporter from an algae species, recombinantly expressing it in the E. coli strain that we use, and providing the synthetic nucleoside triphosphates to the media, and that transporter brought them into the cell and allowed the machinery of the cell to use them. So the idea would be then you'd create a plasmid, we call them information plasmids, they replicate, and then the long-term goal, of course, is that you have to transcribe into mRNA and tRNA. Those tRNAs have to be recognized by orthogonal tRNA synthetases, work at the ribosome to create a protein. Now, in terms of this step right here, Pete Schultz, also at the Scripps Research Institute, and by the way, I think it's a tragedy that he hasn't won the Nobel Prize for this. developed what people refer to as the amber suppression method, where he took a stop codon and then he found tRNA synthetase pairs from a different species. They all wound up being archaea species, where the synthetase would selectively charge the tRNA in E. coli, not charge any of the other tRNAs, and the foreign tRNA wouldn't be charged by any other synthetases. So you would get and then you would evolve that to charge the tRNA with an unnatural amino acid. And then what Pete did was, in addition to that, recode those tRNAs for stop codons, for amber stop codons. So it's remarkable that it works, right? Because it means you're inserting the unnatural amino acid at every spot where there's an amber stop codon. That's not quite true because you're completing with release factors, so you probably get enough of it to work. But the limitation there is you only get one amino acid. And the idea we had from the beginning is that we would steal his tRNA synthetase pairs, but not recode them for one stop codon, but use an unnatural base pair to provide essentially an infinite number of new codons that could be used to produce proteins not with one unnatural amino acid, but many, and to also not compete with release factors, and to not have to modify any other part of the genome. That was the idea. So the unnatural base pair that we had, I'm going to skip a lot of work, but this unnatural base pair was the first that we got into cells, and we found that it replicated well. In fact, this is a picture of the cells. These cells are dividing. They're dividing with 6 letters, 4 base pairs. Sorry, 6 letters, 3 base pairs. So since the last common ancestor of all life on Earth, it's been 4 letters, 2 pairs. This organism is growing with 6 letters and 3 pairs. It grew slow, so we published this. What we just published earlier this year, I'm not going to go into this a lot, but it turns out the transporter that we were using was making the cell sick. It was reducing growth rates. So we simply removed the N-terminus of it, because in its native species, that N-terminus was being removed after it got to the mitochondria, which is where it functioned, and it wasn't being removed in bacteria, so we simply genetically removed it. Suddenly the cells weren't sick anymore, so we didn't have to express it on a plasmid. We moved the transporter into the chromosome and explored a lot of different promoters, so constitutive promoters. So there still is a correlation between, this is activity measured by P32 radiolabeled ATP uptake. So there still is a correlation between activity and growth, and so we chose this one right here, which is just only modestly slower than wild type, 42 to 47 minutes or something, so almost no decrease in growth, and a nice robust import of the triphosphate. So, and not only did that make the cells healthier, but it also meant that we didn't have to induce production of the transporters, constitutively induced. They're just naturally competent to take up the triphosphates. And on top of that, it increased our fidelity. From this initial work, the fidelity was about 99.9 per doubling, so maybe 1 in 1,000 you'd make an error. Part of that was coming from the transporter because we were putting it on a plasmid, and plasmid fluctuation copy number was leading to different levels of the transporter. Some of the cells had too little of the transporter, which resulted in too little of the uptake, and they were trying to replicate the unnatural base pair with too little triphosphate. Not a good thing. And so these, with much more homogeneous levels, we could optimize triphosphate addition, and fidelities went up. Now, this pair we had discovered after this, and the initial work in the cell had all worked with this. When we switched to this pair, fidelities again went up, and also, I didn't mention this, but there's some sequences that were not as well replicated, and so optimizing the transporter expanded the sequences that we could use. Going to this pair further expanded the sequences we could use, but here's the problem. This sort of the highest that we were getting in fidelity was something like 99.99%. So when I give this talk to chemists, they're always like, oh, that's great. When I give this talk to biologists, sometimes they're like, wait a minute, 99.99. Don't natural polymerases get something like 99.999999? And do you really have stability if you're exponentially growing these cells? When would you lose the unnatural base pair? So we wanted to try to come in with a system. Well, frankly, we wanted to cheat. And so everyone uses Cas9, so we had to use Cas9. I think it's obligatory in synthetic biology. But we used it differently than we didn't use it for editing. It was actually referred to earlier, what the original response, the job of Cas9 to do in a cell, in a bacterial cell is. It's literally an adaptive immune response. Guides from previous exposure to plasmid or viral RNA target Cas9 for cleavage of when they see that sequence again. Unlike human cells that are so recombinogenic, this doesn't lead to homologs for combination, this leads to loading of a super active endonuclease called RECBCD, and it destroys the element. So we thought, wait a minute, maybe, here's the idea, right? Here's a plasmid with our unnatural base pair. 99.99% of the time it's replicating away nicely and we're preserving our information, but every 1 in 1,000, 1 in 10,000 times it's making a mutation. Could we target Cas9 to that sequence that results from loss of the unnatural base pair? In other words, would the unnatural base pair protect the plasmid from cleavage by Cas9? And indeed it did. So I'm going to only show you one piece of data. We created an information plasmid that had 2 unnatural base pairs, one in an mRNA, one in a tRNA, the 2 guides that targeted the lost sequences. Here's the transporter in the genome, and we've now included Cas9. I'm not going to go through the details here, but you can see this is a crazy growth regimen, right? These number of doublings in liquid, it doesn't even include the doublings that occur during solid plate growth, because we don't know how to characterize that. And you can see in our original system, the fidelity starts high, but then as you grow, you eventually lose it. When you induce Cas9 and you use it to remove the lost sequences by degradation, you see that we virtually can never lose the unnatural base pair. It's present, best we can tell, indefinitely, because anytime it is lost, that resulting plasmid is destroyed. So we think, at least at first order, we've solved the replication piece, and so we wanted to move on to the next part, transcription. I didn't mention this, but during the chemistry phase of development, where we were optimizing the unnatural base pair, we didn't only look at DNA replication, we also looked at transcription. Obviously, we didn't want to create an unnatural base pair that was replicated well but couldn't be transcribed. So we knew that they were transcribed in vitro very well by T7-RNAP. T7-RNAP is what everyone uses to express a heterologous gene in bacteria anyway. So we looked at that. So what you're looking at here, this assay is, we're isolating total RNA from a cell, adding a primer specific for the gene of interest, doing a notoriously error-prone reverse transcription reaction on it, then PCR amplifying it. And one of the unnatural nucleotides has a binding tag on it. So if the unnatural DNA base pair survived that entire process, right? If it survived the replication piece, the transcription piece, the reverse transcription into cDNA, and the PCR amplification, it would have a binding tag and result in a structural evidence shift. And that's exactly what you see here, only when you have the unnatural and the correct experiment. None of the control experiments show the shift. So we didn't spend a lot of time on transcription because we were pretty anxious to get onto this step. So what we did here was, of course, what I'm talking about here is, can we reprogram these orthogonal tRNAs, synthetase pairs from Pete Schultz, not with amber stop codons, but with one of our new codons that we can now store and retrieve by transcription? Can that go to the ribosome? And when you have an unnatural codon with our unnatural base pair, a nucleotide in the message, can it be recognized and decoded by an anticodon bearing tRNA with an unnatural amino acid? So the first thing we did was we actually made 64 different combinations of the unnatural base pair in a variety of different codons, exploring the first, second, and third position. And we did whole cell lysate. We used whole cell lysates to simply ask, could protein synthesis occur, and could they function at the ribosome in a test tube? And so based on those, we sort of ordered the codons as most interesting, and this was one of them that looked good. So what you're seeing here is expression of GFP in a cell, and in this particular case, you're not adding any of the unnatural amino acid. So the unnatural amino acid that we're coding for, that we're decoding, is azitolysine. Azitolysine has an azide, which you can use click chemistry to selectively attach a fluorophore. So what you see maybe a little bit of read-through, but this is the Western, so you see a little bit of GFP, but you see no TAMRA signal, no fluorescence signal, because whatever little amount of protein is being produced, it doesn't have the unnatural amino acid, and therefore it's not fluorescent. The fluorophore has nothing to click onto. If you look at 2 millimolar of azitolysine, but in this particular experiment, we're using natural mRNA. What you see is robust production because it's the natural, but you can see no click because it's the natural. We were pretty happy to see this. If you give it 2 millimolar lysine 3 hours, and you have both the unnatural anticodon and the unnatural codon in the message, you see protein production, and more importantly, you see that TAMRA clicking on, meaning that protein's now unnatural. If you look, you can see, well, maybe there's a little read-through there. So maybe our codon decoding is not perfect. Turns out you can chase it away. If you look, you can maybe see it a little better here, by simply adding more acetylysine. What that means is the inefficiency there has nothing to do with decoding. It's the charging of the tRNA by the synthetase, not the decoding of the unnatural information. We then went to a GXC codon, another codon, same experiment. In this case, you can see that there's a little more read-through in the absence. You can see that really clearly right here in the overlay. But again, when you go to higher concentrations of acetylysine, you completely chase that away. So again, not decoding efficiency, purely charging of the tRNA by the synthetase with the unnatural amino acid. This is evidence that what you're seeing here is the first time information has been stored and retrieved that goes past GC8 and T. So this, we believe, I like this. This is an overly dramatic title to start wrapping up the talk with. But what we have now are semi-synthetic cells, like I just said, that are stably growing, not only with 6 letters and 3 pairs, but are actually storing that and retrieving it in the form of new proteins. And so getting back to some of what the motivations were for doing this in the first place, and in my lab, we're continuing to explore the potential sort of like conceptual ideas, and also some of the more practical ones. But I also, in 2014, started a small biotech startup called Syntherex, which stands for synthetic organism. We probably should have called it S-Syntherex for semi-synthetic organism. But that company's now got 13 people working at it. It has 2 pharma deals, both of which have been first phase completed and are now moving on to a second phase with the pharma to produce therapeutic proteins that are of particular interest to this pharma. And the Syntherex also has its own core pipeline producing proteins with modified unnatural amino acids for different therapeutic applications. So I think I've spoken quickly and actually moved through my talk faster than I thought, so I'll slow down a little bit because this is probably the spot I should slow down. This is my group. The people in red are the people who work on the unnatural base pair project that I just described. Let's see, Emil and Rebecca are first-year grad students. Vivian and Sydney are second year. Aaron and Michael are third year. And York Zhang is a fifth year, who is an extraordinarily talented graduate student from Canada, actually. He did all of the translation work that I just described. And I'd like to thank these agencies for funding, and again, you for both the invitation and for your attention.
Yes, you can stay there, and we have some around 15-20 minutes for questions. So, any burning questions?
I had a question, and I'm sorry if you said it, I missed it. Are the tRNAs also encoded within the cell?
Yeah. Oh, yeah.
So it is now independent
Yep.
of external?
Nope.
No?
We have to add the triphosphates,
Aha, okay.
and that might fit with some of the discussion that we may have later, because we've done lots of experiments in my lab where we don't provide the triphosphates, and the cells cease to grow. Well, so I've got to be clear. If we have one unnatural base pair, they grow really slow, and then they mutate it out and grow. We've never done that experiment with Cas9, so maybe they would die. But even just one, without Cas9, they grow eventually. With 2, even without Cas9, they can't do that. So it's just probabilities, right?
Okay, thank you.
One question to start with on my side. So you're calling these new base pairs unnatural base pairs?
Sure, unnatural.
So yeah, and you clearly say at the beginning this, for some reason, whatever reason it is, evolution didn't select for these, so we have the 4 natural base pairs. So you think that introducing those new ones, and you're able to show us that you can replicate them, they can transcribe and all that, would evolution keep some of those new ones, or how will evolution in general, do you think, evolution could accept these new?
So evolution cells cannot make our base pairs. So let me be clear, this is not a Jurassic Park scenario.
Yeah, exactly.
Because getting back to what I said very early about the fact that they're totally synthetic, right? These have been made in my lab and now in China and no place else. The conception, the possibility of nature evolving the biosynthetic pathways for these triphosphates is zero. That's the probability they will do it. I mean, just for fun, I'll note. I mean, look, evolving an entire set of an entire new pathway de novo. That's just not the way nature works, right? Evolution works by tinkering. Works by taking something close and sort of exapting it to a new function. And if you look at the analogs that we're using, for example, this particular analog right here, it's what chemists call a C-glycoside. Nature would have to use electron transfer to make that. Maybe it could, cobaltamine sort of cofactor chemistry, but that looks wildly difficult for nature to make, to forge that carbon-carbon bond. There's just nothing in nature that looks like these nucleotides, so the idea that they're going to sort of magically sort of mutate the ability to do so, let me put it this way, it's not a single point mutation away. Now that gives us the advantage to whatever extent that you're worried that these bugs are going to escape and grow into Godzilla and kill Tokyo. You know, like I said, I guess you could consider this a kill switch, right? You stop providing the triphosphates, they stop growing. And you know, what I like most about this is that those are our triphosphates, right? We made them, we designed them, we built and tested them from that rubric. And I love having spent all these years on the chemistry and now working on the biology. So one of the things we're doing now is working on understanding what a DNA replication fork does when it encounters the unnatural base pair. And I think there's some really cool biology that's coming out of that. For all you maybe bacterial recombination aficionados in the audience, loss is 100% mediated by Rec A recombination. You delete Rec A, you don't need Cas9.
Yeah, we have a question.
Somebody else has one on.
Aww.
Okay, yeah. So I was wondering if you could tell us a little bit more about the properties of these as bases, and do you know, for example, if you have a run of them, do they form a normal sort of B-form helix, and how stable are they compared to hydrogen bonding? And that kind of
So if you put in one unnatural base pair, this particular one right here, it's as stable, it's slight in one particular sequence context that we examined, because all this is sequence context. In one particular sequence context that we examined, it was slightly more stable than an AT, less stable than an GC. That was one. If you put in runs, you start to distort the duplex and you lose stability. And the reason is because, so I usually go into this in the chemistry version of this, the pairs in duplex DNA don't do this, right? There's nothing to be gained from doing that. What they do is this. And they do provide a small local distortion. What's really neat, the best part of my chemistry talk, is that seems like that should be a problem, right? Because that looks like a mispair. Polymerases don't synthesize mispairs. So it turns out that during synthesis, when the polymerase grabs the triphosphate and pairs it against a template, they do this. So they take instruction from the polymerase. But once they move out of the site, they do this. So that opened up all sorts of other questions, which we've now answered, which we haven't published yet. They aren't repaired by NER or BER. MMR does monitor them as they come out of behind the beta clamp and the replication fork. From our perspective, this is probably the single thing that I like most of any result we ever got. MMR fixes mispair. It selectively, when you delete MMR, fidelity goes down. So what that means is MMR is recognizing our pair at least somewhat better than pairs of our unnaturals with the naturals. So it's kind of recognizing at some level our unnatural base pair as a bona fide unnatural base pair. In terms of not being recognized, so not being recognized by BER is no surprise, right? The endonucleases aren't going to be able to grab those. There's nothing to grab onto. NER, what we were obviously worried about, they don't cleave them, they don't remove them. If you delete NER, you get no effect whatsoever on retention. And it might again either suggest that the duplex isn't distorting enough, or that, it probably suggests that.
More questions?
Uh,
Rachel?
So
I don't know if I fully understand this, but is what you're heading toward, could it be described as like a chassis for SynBio that would have increased functionality because of the base pairs?
Sure.
Okay.
So I tend not to like, synthetic biology is very, very impressive with itself. In an odd kind of way, I've never considered myself a synthetic biologist. I always considered myself a chemist. And for the first 10 years of this project, we really did medicinal chemistry. That's really what it was. It was making small molecules, studying their solubility, studying their uptake into cells, studying their off-target activity, studying constant dose-dependent effects on what you were looking at. Very much analogous to what MedChem does. Synthetic biology comes along and wants to now define things as, I don't know, think, build, test, whatever that mantra is. It's not exactly clear to me it's all that different from doing good genetic engineering, but I don't want to get into that fight. The term chassis, sure. Okay, we now have a cell line that can express proteins with, we're only up to 21 amino acids. But we have every reason to believe that we could do 22 right now, because there are 2 pairs of orthogonal synthetase pairs. What we're going to have to do next is start to develop, we can no longer use the field's tRNA synthetase pairs, because 2 is all they have. No one's ever had motivation for getting more than 2, because you only had 1 codon. And there are interesting ways that we can start to use the unnatural nucleotide as a recognition element to mediate specific charging. That would be really cool. But sure, it's a chassis. So if I want to use that language, and it would probably help me get papers published.
There was another question.
I wanted to just ask a simple question about the possibility of these unnatural base pairs forming bonds with the natural ones, as sometimes happens. I guess the non-canonical type in
Like a
RNA.
slip or something? Or
Like a wobble pair, for instance, in RNA.
a wobble still has hydrogen bonds.
Right. But so is there any basis for imagining that this could ever happen with an unnatural engine?
We've done lots of just thermodynamic studies and tons of kinetic studies. Our biggest problem tends to be A, mispairing with A, for obvious reasons. This is the most hydrophobic of the naturals, and it can most tolerate losing waters. Because what's happening when you pair a triphosphate, an unnatural triphosphate against its unnatural in the template, you get a very strong hydrophobic effect driving that. In contrast, when you pull out a natural triphosphate, in solution, it's solvated by waters. So to pair it with a natural, you either have to majorly distort it so that it can hold on to the waters, and even that's bad, right? Because you're essentially taking water from an aqueous environment and putting it in a hydrophobic environment, which it hates. Or you're desolvating it, which is another penalty. So that hydrophobic effect is probably a big part of what prevents this. But we think that A is simply best at cross-strand intercalating. So A tends to be the biggest mispairing problem kinetically, and they tend to also form the most stable thermodynamic pairs thermodynamically, the most stable pairs thermodynamically. And so I think, so I didn't really go into this, but choosing the hydrophobic effect from the beginning was to try to get us away from mispairing with the naturals for this exact reason.
So I would like to maybe switch a little bit from the very cool science into applications. You've been involved in 2 companies. One had an IPO in 2014. The other one was acquired by Roche Genentech. So tell us a little bit about your experience with those and what's the commercial potential for these? Or are these, basically, tell us the commercial potential.
Okay, so those 2 companies that you just mentioned are not Syntherex. The Syntherex was a third company that's based on this technology. Those 2 former companies, one was a company called Kaogen. It went public in 2014, AKAO, if you want to buy stock. Good deal. They actually so they're both, so it's an antibiotic company. It has a novel aminoglycoside called plasmycin. Just completed 2 phase 3 trials. Both look good, and it is almost certainly going to be FDA approved sometime early next year. And most importantly, it completed a trial on a Cree trial. If anyone follows the antibiotic literature. Cree is this really, really scary bacteria. It's carbapenemase resistant. It's tended to be Klebsiella in the early days. It's also called the New Delhi strain because it came out of India. The reason it's so scary is because it had a beta-lactam. For years, beta-lactamases, the one thing they couldn't cleave was carbapenemases. Were carbapenems. Cree is a carbapenemase, so it cleaves imipenem. So imipenem was something that was very important for use against M-insensitive plasmamycin, has now activity against Cree, those strains. It is going to, I think, generate a lot of interest. So I started that company in 2004. The second antibiotics company was a little company called RQX. Yeah, it was a weird experience. That company only existed for 2 years, and then Genentech Roche bought it. It's currently, the molecules that came out of my lab are currently, I think I'm not supposed to say anything, but I'm in Canada, so no. Yeah, it's now the largest small molecule antibiotic development program at Genentech, and I met with the president of small molecule development at Genentech, and he told me that he thinks in 8 months there'll be first dose in human for the earliest trial. So that's exciting. So was that your question?
Well, and I guess your third company that is related to this.
Yeah,
So that was a little bit of a. My question was more the commercial potential for the natural base.
Yeah. So I think the commercial. Yeah. So for synthetic biology. Like I alluded to, I think that the potential for modify. So look, small molecule medicinal chemistry has certain disadvantages. You have to make them. They have the advantages that if you can make them, you can make it. Right? You can use any pharmacophore. I mentioned electrophiles, but there's been all sorts of crazy, weird structures that are simply not available. I mean, look at the beta-lactam. My personal favorite medicinal chemistry molecule are beta-lactams. Really, absolutely beautiful molecules. PK, PD, they just behave beautifully. Atom economy is breathtaking in the beta-lactams. The number of lives beta-lactams have saved is remarkable. You show me an amino acid that's anything like that. The beta-lactam activity is simply outside the scope of what proteins could have provided. Now, I'm a fan of evolution. I think that given enough time and enough search, evolution could come up with pretty good solutions to anything. It's a kinetics problem. They're simply so far away from those solutions. Providing an unnatural amino acid gets you close and lets evolution then fine-tune things. And so we very much plan, and one reason that we're spending a lot of time to make sure that the unnatural base pairs are fully compatible with the cell is because we plan on doing things like phage display, things like selections, things that really differentiate protein therapeutics from small molecules because they have advantages too, even though they can't access certain pharmacophores that chemists can. The protein, the cell does the heavy lifting, it makes the material, you can produce it at high levels, and you can evolve it. That's an extraordinarily powerful property of biological molecules. And so really kind of one way to look at this is we're trying to combine the best of both worlds. The Syntherex, it's now gone through two raises. It's a stable company. It's got 13 people, and they've just completed 2 early collaborations with 2 different pharmas, and both of those on the particular proteins of interest to that pharma, and both of them were successful. And the bar was relatively low. It was just make the protein, show that it behaves, show that it's pure, show that it has the unnatural amino acid content, blah, blah, blah. And so both of them are now entering a more invasive, a more extensive development effort that's sort of like the next step with pharma. And as I mentioned, the company also has its own internal program. I'm not going to tell you what it is because they get all bent out of shape about that, but it's not hard to imagine real obvious ones, right? ADCs, for example, right? ADCs you want, if you were developing an antibody drug conjugate, right, you want 2 ways to specifically attach things, right? You want to be able to attach a drug, and you want to be able to attach something like a lipid or a PEG, right? The drug for activity, the PEG or the lipid for clearance, for PK. You can't attach anything to an antibody selectively, right? Not at one spot, right? So what you use is lysines or cysteines typically, and they're all over the place. Any place you've got an active lysine or cysteine side chain, and that has led to a real problem with the FDA because of homogeneity issues. The ability to site-specifically incorporate one, and kind of most excitingly, explore the site, right? Move it around, and to be able to not only attach one, but be able to attach 2, which we can do now. in which is immediately solves a problem. And so one could imagine that would be a good first thing to think about. But if you just step, what I like most is as a chemist, just think about, okay, if we could expand the repertoire of physical chemical properties available to a protein, and maybe even in the context of an evolvable system, what sort of new activities could we get? So that's kind of what we're most excited about.
Excellent. Maybe we have time for one more question. Yeah, one last question here.
I was wondering whether you use DNA sequencing anywhere in your perhaps validation pipeline, and if so, how do you sequence your unnatural base pairs?
That has caused us not a small bit of problem. Right now, the way we sequence, we have to specifically look for self pairs. So that's when they pair against each other, and the way we do that is there's a variety of ways. For example, you can take a plasmid out of a cell, use that biotinylation trick that I showed, which if you haven't switched, should give you a strand, and then sequence that strand to make sure that's good. That's one thing you can do. We also, and again, this is not actually in this talk. If I can, I'm gonna call up a picture for this in 60 seconds, because I love showing this. As I'm doing this, I'll tell you that I have been contacted by both Oxford Nanopore and Illumina, and both of them are collaborating sort of with my lab. We've given them stuff, and they're applying that, their technology to do this. But I just love this picture I'm about to show you. So if you take a piece of DNA, a plasmid, out of a cell that you've just replicated it, and then you take one strand, you separate them, take one strand, and then you just do a Sanger sequencing reaction. Right? In the Sanger sequencing reaction, there's no unnatural triphosphate. And so what you see when you get, I lied before when I said that was the best day. This was the best day. When we got this chromatogram, the first time we did this, it's kind of like the first time you see the unnatural base pair. So you can sequence along absolutely normally, and then bang. That's the unnatural base pair. And you can sequence the other way. Bang. So we know exactly where it is, it hasn't moved, and it's present. The only thing we have to check is the self pair. We do that as I just described. This is not a particularly high throughput way to do it. It's not that desirable. And we're working on ways. I guess one answer is that, as I mentioned, we're collaborating sort of with Illumina and Oxford. And if one of them come up with a way to do it, we will use it.
We'll see who wins there.
Yeah.
Who's going to put faster. Anyway, thanks a lot for a very inspiring presentation, and it makes us dream about the future. Many things coming up. We will have now a break of about 10 minutes, bio break, and we'll reconvene here for the social and ethical aspects of genome engineering. Thank you very much.